Our sponsor is the Medina Group, led by Dr. Medina. He and his team of postdocs, grad students, and undergrads perform research in the Precision Therapeutics and Bioresponsive Materials Lab. They chemically synthesize peptides to study biomolecular interactions and cellular responses to peptide therapies. Peptides are sequences of amino acids, which are the building blocks of proteins. Peptides are not as large as proteins, but they still serve many important biochemical functions. This function is determined by the structure of the peptide-specific amino acid sequence. Peptides can act essentially as hormones or signaling molecules, meaning they can regulate specific cellular functions or elicit different biomolecular responses. There is a realm of peptide therapeutics in which peptides are being used to create drugs with very targeted treatments and therapies. The overarching goal of our project is to create an application that will allow researchers in the Medina Group to understand the properties of the peptides they made. In doing so, users will be able to both verify that they created the peptide they expected to make and can better understand how to further modify the peptide in later experiments. The current solution within the Medina Lab was an Excel sheet which did not satisfy their needs. The sponsor wanted something that was easier for newer researchers in the lab to navigate and understand their peptides. They also wanted it to be an application that could be run on a device within the lab. The application needed to populate various peptide properties going beyond the capabilities of the Excel file to aid in peptide modification.
The application, Peptide Planner app, allows the user to input an amino acid sequence, and it will use our custom code to produce the peptide mass, charge, and the mass spectrometry. Mass spectrometry identifies and measures molecules based on mass-to-charge ratio, revealing structure and composition of compounds accurately. To generate the chemical structure of the peptide, we have utilized RDKit to take the SMILES code of the amino acid sequence and display a picture of its structure. To do reagent calculations within the Conjugation tab, we have utilized multiple formulas given to us from Medina Lab to calculate the reagent mass/volume and solvent volume. Lastly, when it comes to predicted secondary structure, the usage of the PyPept API allows us to input our SMILES code of the peptide and then output what the predicted secondary structure of the peptide would be. This all allows Medina Lab to be able to utilize the Peptide Planner app to verify the samples that they are creating within the lab, as well as help to get newer students up to speed. From this, we have completed our project goal of creating an application that can allow researchers within Medina Lab to understand properties of the peptide made. This allows the user to verify their created peptide and to better understand how further modifications can be made. Since our project was completed earlier than our final deadline, we had the ability to even test our application to get first-hand user feedback in multiple sessions, which greatly improved our final design.